Hello and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane and this is episode 143 for the podcast. This week, I had the good luck to sit down with MoonLake Chief Scientific Officer Kristian Reich. MoonLake is a clinical stage company leveraging nanobody technology to develop medicines for immunologic diseases. Kristian, the co-founder of MoonLake alongside CEO Jorge Santos da Silva, has more than 25 years' experience in dermatology and immunology and has been described as a global clinical leader in skin and immune disease. In our interview, he explains how he came to found MoonLake, how the company came to license a key molecule from Merck KGaA, and the impact that their nanobody technology is expected to have on patients in the near future. I hope you enjoy this discussion with MoonLake's Kristian Reich. So Kristian, welcome to Beyond Biotech.
Welcome, Dylan.
Maybe you can start by telling us a little bit about yourself. You've had 25 years of experience in dermatology, immunology. What got you started in this field?
Yeah, I love chronic inflammatory diseases. I have to say, as an immunologist, it's fascinating to think that one of the most delicate systems we have in our organism can give you health and needs to protect us against all these microbial pathogens that we live in. But on the other hand, that you see that exaggerations of certain immune responses really drive chronic inflammatory diseases. And if you look at chronic skin diseases like psoriasis, atopic dermatitis, you have them in the gut, Crohn's disease, ulcerative colitis, we have rheumatological chronic inflammatory conditions. If you lump them all together, that's, a prevalence of 20-30%. So it's a very relevant proportion of the chronic diseases that human beings can suffer from. And they are obviously driven by something going wrong with the system that ultimately also has a very strong and important physiological function.
Now, you saw all of those things as a clinical leader, as a clinician, but was there a particular moment that inspired you to jump out of the practice and then move into co-founding MoonLake?
Yeah, Dylan, what you can see, but the listeners cannot, I'm old, right? So I started in medicine many, many years ago. When I started in dermatology, Dylan, believe it or not, we had topical corticosteroid creams and tar and phototherapy, and it was a little bit frustrating. So we had 200,000 names for 2,000 diseases, but we had 3 treatments. I'm overdoing it a little bit, but not much. And then came the fantastic era where we began to understand our diseases. So suddenly I can say, wow, psoriasis, we have T cells in there. We know what activates these T cells. We know what mediators that these T cells make that probably drive the disease. And then came this fantastic moment, and that was not me, unfortunately, but where it became clear that we can manipulate antibodies, so a molecule that all of us have, molecules that have a super high specificity and sensitivity, Dylan, in the sense that one antibody wants one and only one molecule only. Now imagine you suddenly can use this toolbox to create antibodies that specifically inhibit these mediators. And that's exactly what happened more or less at the beginning of my career in dermatology. So suddenly we had these antibodies. We could inject people with antibodies that would normalize these mediators that we had identified to drive the diseases. And I take psoriasis as an example where patients were 2-3 months hospitalized every year. Tar was put on their skin. They were getting some phototherapy. They would get better a little bit, and then they would come back into the hospital. And suddenly we could inject 1 ml of an antibody, and half of the patients would clear. So I think I was lucky because at the beginning of my career, and you said it, I'm an immunologist and dermatologist, but I saw this translational revolution, Dylan. I saw how our insights into immune mechanisms were converted using this targeted therapy strategy into treatments that would completely change the way we could manage some of these important conditions. And that really got me going, right? So I did a lot of research. I participated in clinical trials. I think I can say I helped many of these targeted therapies to become reality. And then Dylan, at a very much later point in my life, suddenly there was an opportunity to look at a different kind of molecule. I would call this targeted therapies 2.0. So no longer we are now talking antibodies, we are talking nanobodies. And of course, having really seen every day for many years how antibodies transform treatments, and now thinking that we have an opportunity to do what antibodies did just better with this nanobody sonelokimab we could get in our hands. That for me was the real jumping point. Why don't you, at the end of your career, why don't you now take this opportunity and all your knowledge and all the science and try to develop something that is even better than the targeted therapies we had before?
What's interesting, and I'm sure it's going to come across to the listeners as well, is you seem still really excited about this. The science has still got you very excited.
Dylan, I am still excited, right? This is not played. And look, I have been a real physician, a real dermatologist for many, many, many years. I've seen thousands of patients with chronic inflammatory diseases. Because we share some of our treatments with other specialties like rheumatology or gastroenterology, I'm also very aware of the chronic inflammatory diseases that sit in these specialties. I still think that these patients have an enormous amount of suffering. Think about it. Many patients, I take again psoriasis as an example. It starts when you are 16, and you know that you have 60, 70 years ahead living with psoriasis. So every company and every doctor will say unmet need and burden of disease, but I think I experience every day for many years of my life what it means to have a chronic inflammatory condition. And just imagine, and also having a little science heart, what it means that suddenly you have a completely new drug class in your hand. And then an additional excitement thinking that you may contribute to developing a new therapy that kind of sits on the shoulders of these fantastic targeted therapies that we saw becoming reality in the last years. So why shouldn't you be excited? Have we solved our chronic inflammatory diseases? Can we cure them? Do we have treatments for all of these conditions that make 90% of the patients 100% better? No, right? So there is still a lot to be discovered. And my excitement is driven by the thinking that with our nanobody, that with sonelokimab, we could do it exactly this.
Let's talk a little bit about MoonLake Immunotherapeutics. What's it all about? What's the core mission? What gets your team out of bed in the morning?
So this is a company that is created around one molecule. You heard me talking about this before. It is this nanobody sonelokimab, and I will explain in a minute what this molecule is. There was a time some years ago when there was an opportunity to get full access, full rights, the IP, the molecule, everything. It's coming from the German Merck. We were able to organize an exclusive license agreement with German Merck. And together with Jorge Santos da Silva, there was a famous moment at the airport in Frankfurt where we decided, wow, if we can get this molecule, let's go crazy. Let's leave our comfortable lives behind. Let's get the investors on board. Let's form a company. Let's develop the strategy. And this is what happened.
You're focusing on a couple of very specific diseases in there and very serious diseases as well. Is there a reason that you've chosen those specific inflammatory diseases?
There is a reason, Dylan. And to oversimplify, but just imagine the world of chronic inflammatory diseases as certain immune programs going wrong. You look at your washing machine, I've been told that there are programs for silk and for cotton and for 90 degrees and 100 degrees. And similar to this, our immune system is able to pull off different programs. For example, to defend against bacteria or fungal infections or viral infections or kill malignant cells. And all of these different programs, they have to be in balance. And today our understanding is that an exaggeration of certain of these immune programs drives certain chronic inflammatory conditions. And there clearly is a group of chronic inflammatory diseases, Dylan, that are driven by too much of a cytokine pathway that is called interleukin 17. And there has been a lot of learning. We now know that it's not one cytokine, it's actually a family of cytokines, A, B, C, D, E, F. We know that two of these family members, namely IL-17A and F, they cooperate to drive a certain group of chronic inflammatory diseases by forming dimers, right? So you have an AA, an AF, and an FF pair of these cytokines that drive diseases. Now, our nanobody blocks IL-17A and IL-17F. I think this was one of these exciting initiating events, that this is obviously an MOA, a mechanism of action that would allow us to better than before inhibit the IL-17 pathways with a perspective to treat diseases that are driven by an exaggeration of this IL-17 pathway better. Number two, Dylan, antibodies are relatively large molecule. We measure the size in kilodalton. And to give you a feel, a normally occurring antibody in all the therapeutic antibodies, they have around 150 kilodalton. Our nanobody has 40 kilodalton, so it's only 30% of the size. And there's good evidence that with smaller size comes a better tissue penetration. So Dylan, if you have a skin disease, I inject you with a nanobody that's absorbed into the peripheral blood, but then it has to go where the inflammation sits, in this case, the skin. And we think that being smaller as a nanobody, we have a better chance to get more of our peripheral blood molecules into the skin. There's another advantage. With our nanobody is actually composed of 3 binding blocks. With 2 of them, we block the 2 cytokines, IL-17A and F that I was talking about. With a third, we bind to albumin, so a naturally occurring protein. This was introduced to prolong the half-life to the 2 weeks that we have, allowing us very convenient monthly injections to keep diseases under control during maintenance therapy. But inflammation, Dylan, comes with an increased permeability of blood vessels. And this increased permeability leads to something that doctors call edema. So fluid with albumin leaves the circulation and accumulates at the sites of inflammation. And guess what? And that's not rocket science, but if your molecule binds to albumin, what a traditional antibody cannot do, it should steer our molecule selectively to where the inflammation sits. So helps us to specifically enrich at sites of inflammation. So the molecule characteristics would indicate that we would be particularly working well, A, in diseases that are driven by IL-17, but B, in diseases where the inflammation is difficult to reach. And when we looked at chronic inflammatory conditions that are driven by 17 and that are underserved and where antibodies have struggled, a chronic inflammatory disease called hidradenitis suppurativa came up as one important disease where all these particular characteristics are the reality. And this is why we picked hidradenitis suppurativa as one of the first big indications that we would go to. Psoriatic arthritis is another one, a disease that sits in the skin, in the nails, in the joints, in parts of the joints that we call the enthesis, so where tendons, ligaments, capsules insert into the bone. I would also call this a difficult to reach site of inflammation. So if you think about it, we did not pick our indications randomly, but they were carefully selected for making the most out of the specific molecular characteristics that we have. And we continue this path. Also, the new indications that we are looking at, I think they're all indications where you see this pattern.
You mentioned getting that molecule licensing deal from Merck KGaA. That's got to be a milestone in getting the company off the ground. But have there been other milestones that you can point to in the last couple of years, things that have really stood out and made MoonLake bigger and better?
Well, yeah, absolutely. I mean, the company was formed in 2021 and then 2022. We went public, Dylan. We were all celebrating at NASDAQ, celebrating with a tear in the eye because not long before the Ukraine war had started. So the world was, had many question marks, but there we were in New York, Times Square, and going public with our company. That allowed us to raise a significant amount of money. We already had a phase 2 trial in psoriasis, but this money allowed us to then go into the indications, hidradenitis suppurativa, psoriatic arthritis that I was talking about. So that would be a second important milestone, going public. And the third, Dylan, was, and for me particularly exciting as the Chief Scientific Officer of MoonLake, when we saw the first result in hidradenitis suppurativa. The small size, the preferential accumulation at sites of inflammation, the special way by which we block IL-17A and F. Dylan, this is all stories. Ultimately, you have to show that these stories convert into better outcomes. But that seemed to be the case. So there was a moment when we looked at the results of our big global phase 2 in hidradenitis suppurativa, and we could, I think, really show a response that was so far unseen, unheard of. And back on the back of this data, we were able to raise another another $500 million, then allowing us to go into phase 3. And as you're probably aware, so this would be my personal third big milestone. And as you're probably aware, later this year, we will be able to talk about the phase 3 results in this indication and discussing with the world the primary endpoint data, really to prove if all these molecule characteristics, this great data in phase 2, if this is now all culminating and replicated in phase 3.
Let's dive into a little bit of that science then, and maybe we'll start by just taking a step back. Assuming I know nothing about nanobodies, where can we start? What you've told me so far is that they're significantly smaller. So instead of 150, they're down to 40. I mean, that comes with the name. What else makes them different to a traditional antibody?
Dylan, and I could clearly chat for 2 hours about these molecules. They were, I think, discovered by serendipity. I had the chance to have dinner with the person, a Belgian scientist that actually discovered nanobodies, I believe. These are derived from antibodies that only have a heavy chain. You must understand that if I extract an antibody from your blood, Dylan, I will find that it's composed of a heavy chain and a light chain. And the variable regions of both these heavy and light chains, they ultimately dictate what specific target your antibody binds to. Now, and I think this was in the times when there was the peak of the HIV crisis, and the students that were about to investigate immunoglobulins in human blood, they said, we don't want to work with human blood. So they opened the fridge and took out some camel blood. And they saw bands that they couldn't allocate to any known immunoglobulin band. And they could see, and then later it was discovered, it's not only camelids, it's sharks, some other animals that really have antibodies that are different. They have no light chain. They only have a heavy chain. And it's this variable region of a heavy chain only antibody, Dylan, or a VHH that is really this nanobody. This is only 15 kilodalton. But one beauty of these nanobodies is that you can easily create multimeric molecules, molecules that bind to different targets. And sonelokimab is a shining example. So ultimately, if you really technically look at this, sonelokimab is covalently linking 3 of those nanobodies. And this is where these 3 binding blocks come from. With 2, again, we bind IL-17A and F, and with the third, albumin. So this means nanobody, a variable region from a heavy chain only antibody, 15 kD, sonelokimab, 3 nanobodies, if you will, covalently linked, 40 kD, but still enjoying the advantages of the molecule that I was discussing earlier, including the smaller molecule size.
You know, I love those serendipitous stories, and it just seems amazing to me that you could just open the door in a lab refrigerator, find something from a camel, a llama, you mentioned a shark as well. Was it really that simple, or is this just a story that we tell?
Of course, it's the story that we tell, right? And I'm sure that it was a lot more complicated. But I think some crucial elements are really true. And Dylan, we just have to embrace that some of the key discoveries in human science, they are made by serendipity. And if you ask me, I think the era of nanobodies is just dawning. They are already used to be labeled and to help, for example, to diagnose certain malignant tissue in patients. They do this with their better tissue penetration a lot better than antibodies. I can see that they are being used to create passive immunization. There are, for example, nanobodies against the coronavirus underlying COVID-19, and they work a lot better than antibodies that you can give to passively immunized patients. They seem to not only penetrate tissue better, it also seems that they bind certain epitopes in their target that are very special. I give you an example. If some tumors you can treat by blocking a growth receptor on these tumors by giving an antibody, right? Breast cancer, for example. But the tumors are unfortunately clever, and mutations will occur in these growth receptors, and then the antibodies no longer bind. Nanobodies against the same growth receptor still bind. So there seems to be also a reason why evolution has brought about nanobodies. And of course, we would be very proud if we could take this present of nature to treat chronic inflammatory diseases better.
Can you talk about what's actually happening at the molecular level with these nanobodies? What they're doing to the patient?
Yeah, and let me do this very specifically for sonelokimab, right? So just imagine, and then now I take hidradenitis suppurativa. It's a terrible skin condition. It's one of those conditions that start often in puberty or in early adulthood. Patients develop inflammatory nodules, inflammatory abscesses, very, very painful lesions in the skin. And because there's a lot of tissue destruction and the hair follicles, they rupture and scars form and also tunnels. So structure that have a lumen that shouldn't exist suddenly form deep in the dermis. They fill with pus. These tunnels grow. They can get access to the skin surface. And then really the pus is oozing out of your body. So you have painful inflammatory lesions that turn into irreversible scars. You have these tunnels forming. There's an odor from the secreted pus. So it's clearly a disease that is very hard to see how you can lead a normal life with this, and the disease burden is massively. If you take affected tissue and you ask the question, which cytokines, which mediators are apparently and exaggeratedly expressed in these lesions, you find IL-17A and you do find IL-17F. And if you take certain target cells, keratinocytes, Dylan, these are cells that form the outer layer of your skin, they react to IL-17A and F. If you give a little sprinkle of IL-17A and F to keratinocytes, they make many mediators, and now it comes, that attract more immune cells into the skin. You see that IL-17A and F, they seem to be sitting in the middle of a vicious circle that can explain the extreme chronicity of the disease, that can explain the tissue destruction. Now, just imagine what you now have to do is you have to block the biological activity of IL-17A and F. And ideally, you do it by blocking these three dimers that I was talking about. So if you now come to me and I inject my nanobody, I will do it in your subcutaneous tissue. 1 ml. It's a short 3-5 second long injection. That's all. I will inject 1 ml in your subcutaneous tissue. From there, it will be absorbed into the peripheral blood. From the peripheral blood, it will go back into the tissue, in the skin, where the inflammation sits. And it will bind to IL-17A and F so that these cytokines are inhibited. They're no longer biologically active. They can no longer activate keratinocytes. Keratinocytes no longer make these mediators that bring more immune cells into the skin, and thereby you can put a break into the inflammatory process. You can see the nodules, the abscesses disappearing. We can see the tunnels stop draining. We cannot make tunnels immediately go away, but we can make them stop draining. And then, of course, the symptoms also go away. The pain goes away. Patient can again go to the gym. So the idea is we need to have these molecules that very specifically, like a laser sword, go to the core of the inflammation. And in this case, the core inflammation is to block. And the core of the inflammation is IL-17A and F, and the therapeutic modulation would be to block the activity of these two cytokines.
These particular inflammatory diseases that you're talking about, the psoriatic arthritis and the others, how prevalent are they? What sort of caseload are we talking about?
Very prevalent. And we describe these diseases, Dylan, today as the so-called type 3 diseases. Type 1 would be a disease like lupus. Type 2 would be a disease like atopic dermatitis. And the type comes from an exaggerated expression of a specific cytokine. So now type 3 would be the disease where you find too much IL-17A and F. That's the joining element of these diseases. Psoriasis, Dylan, 2 to 3% in the Western countries. Hidradenitis suppurativa, probably similarly prevalent, 1 to 2%. Psoriatic arthritis, 0.5%. And then there's a growing list of diseases that also share as a main pathophysiological element this overexpression of IL-17. So I would think that altogether, the chronic inflammatory diseases in this type 3 bucket could be easily 10%, 15% or more. And single individuals of these diseases alone are already very prevalent conditions.
Now, obviously, you're talking a lot about size when you're talking about nanobodies compared to traditional antibodies or other antibodies. What does it actually mean for the patient? Is it just that 1 ml injection? Is it a smaller injection?
I think convenience does play a role with chronic therapy, Dylan. Let me start by quickly talking about tablets, right? Because that's a question we also often get, right? Are there tablets? But I have to say, my experience is that patients enjoy having a 5-second shot once per month compared to taking tablets twice or once every day, right? If I have to take antibiotics for 3 weeks, probably I forget it on 7 days, right? It's you travel, you go outdoor, you forget your tablets. So I think just to start with, I think that the subcutaneous easy delivery of these molecules is a great advantage when it comes to convenience. Now, the nanobodies are very small. That means that you can deliver the therapeutic amount, the volume that you need to deliver is small. In our case, it's 1 ml. These drugs are very small, so the fluid has a low viscosity. It's very easy to inject this molecule. As I said, our injection is only 3 to 5 seconds. And then we haven't talked about this. These nanobodies, at least sonelokimab, they also do not seem to develop a clinically relevant immunogenicity. You have to understand that whenever you inject a protein into a human being, there is a risk that this human being develops antibodies against this drug. The important question is, do they impact the effect? Do they reduce the drug levels? Do they reduce efficacy or safety? And with sonelokimab, we also do not see a clinically relevant immunogenicity. That means you can have your short, easy, painless injection, 5-second once per month, and you don't run the risk that you lose the response. This is at least what we have been seeing in our clinical trials. Very stable response over time. And on the other 29 days and 23 hours and 59 minutes, you can basically forget about your disease. So I think some of these features are shared with antibodies, but I would think compared to the antibody treatments that we have in HS, this better convenience that we think is linked also to our molecule, the nanobody, is already an advantage we have in our pocket.
Tell me a little bit more about what you're seeing in the clinic. You said you're moving into phase 3 trials.
Yeah, absolutely. So let me focus to start with on hidradenitis suppurativa. We did a large global phase 2 in 234 patients. This was done in North America and in Europe. You often, Dylan, when you start treating patients with very active disease, so they have high levels of the mediator that you want to inhibit, you need to give a little bit more in the beginning. This is called an induction therapy, and then you move on to a maintenance therapy. With our drug, the induction therapy goes to week 8, and already from week 8 on, that would be every other week injections, we go to monthly injections. That is exactly the regime we tested in this phase 2 trial. And by the way, this also compares favorably to some of the antibody induction regimes that go a lot longer, to 16 weeks. We have a lot more injections to get the drug going. In this phase 2 trial in hidradenitis suppurativa, we tested different doses of our molecule. We have seen, Dylan, that compared to psoriasis, a more superficial skin condition, antibodies often need to double their dose to see some efficacy in hidradenitis suppurativa. I think indicating again that antibodies struggle to a certain degree to reach these difficult to reach sites of inflammation. The dose that we saw being optimal in HS was the same dose we saw to deliver an optimal response in psoriasis. We also had, and of course you compare your drug to placebo to really show what it's doing, but in all the phase 2 trials that MoonLake has been doing, we always have an active reference arm. So we also use a drug that is known. We use a drug that has already done phase 3 trials. Why do we do this? Not so much to directly compare, but to validate our findings. If you can replicate the data that others have been shown previously with a drug that is around, I think it's telling you you got your study design right. You have enrolled the right patients. So to summarize, we did this 24-week trial. The primary endpoint was at week 12. We used a higher outcome than others have used before, an outcome called HiSCR 75. The number indicates the percentage improvement compared to baseline, so at least 75% improvement of HS. We saw a delta to placebo of, for our 120 milligram dose, Dylan, of 29%. In similar trials, the existing antibodies have shown numbers 12, 16, 18. So I think now you understand why there was a lot of excitement. It really elevated the bar. It did this with a very convenient induction and maintenance regime, really also decreasing these deep dermal lesions. We saw specifically tunnels stopping to drain. We saw abscesses melting away. And then we saw a very, very convenient safety profile. Responses didn't stop at week 12. We could see that they would further continue between week 12 and week 24. And based on this data, we said, let's do a phase 3. We could use the same study design. We did our phase 2 pivotal like, Dylan. If you discuss with knowledgeable people, they will always tell, yeah, but you run a high risk that your data between phase 2 and phase 3 is kind of being compressed. Yes, there's always this risk, but of course your risk is a lot smaller if you do your phase 2 properly, if you do it large, if you do it placebo and active reference control, if you already use a study design that you plan to use in phase 3, if you use the same dose, if you really have identified your optimal dose in phase 2. So based on the shoulders of this very exciting and successful phase 2, Dylan, we designed our phase 3. This was started in May 2024. And as I said earlier, we expect to read out the primary endpoint, which is week 16 in this case, later this year.
I guess that the trial design is really important, but then there's also the business side too. Two areas of operations I'd like to hear you talk about. One is manufacturing, so being able to produce the nanobodies in the amount that you need for the trials. And the other is any regulatory challenges that you're facing, manufacturing and regulation.
So regulatory is a beast on its own, right? If you would ask me, Kristian, did you ever dream about regulatory when you were a dermatologist and immunologist? Not so sure, but there are good reasons why this is probably the most formalized, regulated product development that you can do. You treat diseased patients. You have to do this in Europe and you have to do this in the US. The regulatory authorities not always overlap. So believe me, there are a million of regulatory hurdles you have to jump over. But I think it was recognized that we did our phase 2 studies properly. The FDA and the EMA both agreed that we had identified the dose clearly in phase 2 with an optimal benefit risk profile that allows us to only do one dose arm in phase 3 compared to placebo, so we can really simplify our design a lot. And I think we have found good arguments for most of our cases with regulatories east and west of the Atlantic. So I think we are extremely confident about our interactions with the regulatory authorities. Of course, you also have to be clever, right? And it doesn't mean that you should do everything like others have been doing it. Again, we moved the outcome from HiSCR 50 to HiSCR 75, but regulatory authorities recognize this. They see that you don't want to just do a me too product. They see that you really want to challenge your product. Can you get more? So I think this was pretty much appreciated. Manufacturing a completely different animal. Nanobodies are not made in Chinese hamster ovarian cells, the typical host cell to make antibodies. They are actually made in a yeast cell called Pichia pastoris. The process, however, is extremely robust. We think that there's even some advantages when it comes to COGS, for example, down the line. But we have inherited this process from Merck. It had been transferred several times. We inherited an already extremely stable process. We are very happy with the site that is doing our drug substance. We have already produced everything, every drug that we need, not only for our ambitious clinical program, but even for the first years after launch. So I think also manufacturing-wise, we're in extremely good shape given the stage of the company we're in.
And are you working with other pharmaceutical companies, or it's just Merck through that licensing deal?
We are working now with no other company, right? We work as MoonLake, left, right, and center. Merck got some money, obviously. Merck got a certain percentage of MoonLake in the beginning, but this is where it ended. We had a science collaboration where Merck helped us to understand how our model, how our nanobody would look like in an environment where all the dimers and albumin is around, and they helped us to clearly document that sonelokimab can bind two IL-17 dimers and albumin at the same time. But I would think, Dylan, other than that, it's all MoonLake now, 110% MoonLake. There's also no cooperation with other pharmaceutical companies. We know what we are doing. I think we have proven that we can develop, that we can do our clinical development in a very fast and very valid way. I would think that when we fully enroll our phase 3, we have done it with at least double the industry standard. We take full advantage of being a small, aggressive, lean biotech company. But at the same time, of course, we really open up to every quality measure that you can think. If you look at the design of our phase 2 trials, I think you will recognize they're all done in an extremely solid way. We did a lot of basic science. We did a lot of activities to understand the characteristics of our molecule better. So I think MoonLake has proven that it can create a scientific, a manufacturing, a clinical operations package that would be a challenge to be pulled off even by big pharma in this relatively short period of time.
Let's take a look forward now. If we can just look forward generally when it comes to nanobodies, where do you see them developing just as a subset of these biologicals in the years ahead?
I said earlier, Dylan, that very clearly the antibodies have been an absolute therapeutic revolution in the space of immunology and inflammation. They have also been a revolution in oncology, but let's focus on inflammation here. Most of the antibodies that we currently have, they block one cytokine and one cytokine alone. But there is increasing understanding that many of the chronic inflammatory conditions are more complex, that they are driven by more than one pathway. And I said earlier that one beauty of the whole nanobody platform of nanobodies is that you easily can create multi-specific molecules. So as I said, I think that in 5-10 years looking back, there is an era of nanobodies dawning. I think we will see more nanobodies coming. We will see more nanobodies coming that inhibit more than one cytokine. And again, sonelokimab blocking IL-17A and F is a first shining example. I think there are more to come. I do think there is more and more solid evidence that the story that I was telling, the better tissue penetration, the enhancement at sites of inflammation, the low absent, I should say, clinically relevant immunogenicity, probably some advantages in the way they are manufactured. I see multiple reasons why nanobodies can easily become the antibodies 2.0, and not only in immunology and inflammation, but definitely also in immunology and inflammation.
If that's nanobodies generally then, but where does MoonLake fit? Can we look a little bit forward? I know you've got some results coming out relatively soon. Where are you going to fit in this world of nanobodies?
Yeah, I'm telling you right now, I open my eyes in the morning and hope to get through the next day and the next week and pull all of this off. But no, very clearly, we currently develop sonelokimab in phase 3. We do this in adults and adolescents with HS. We do this in adults with active psoriatic arthritis. We have started clinical trials in palmoplantar pustulosis. We have started clinical trials in axial spondyloarthritis. There is a long list of additional diseases that MoonLake may consider attacking. Diseases of your blood vessels, diseases of your musculoskeletal systems. As I said, as we move along, we have an increasing understanding of how many diseases are actually driven by IL-17. So we can extend our sonelokimab play very clearly. Nothing stops us at some point to think about looking at other molecules. Obviously, we have a big passion for nanobodies. But Dylan, right now, we really clearly focus on sonelokimab. We really clearly focus on these important indications in derm and rheum. And we want to first show that we win in those, and then let's do the next step.
Kristian, this has been fascinating. It certainly sounds exciting. If there was just one thing you could leave the listeners with about nanobodies or about MoonLake, what would you like that to be?
Yeah, it's this, I would almost say, don't stop dreaming, right? And don't think because others have been doing things that you shouldn't consider doing things differently. Always in the framework of validity and quality. I think MoonLake is a good example. We have done many things where people were telling us, you can't do that, right? And we did it with success. In the near future, I would say, watch out for the results of our phase 3 trial. We have announced in a press release yesterday that we will talk on the 29th of April in New York about the baseline characteristics of our HS program, taking obviously another variable out of the game. We will announce exactly when we will share with the world the data from the primary endpoint, because the studies will continue, but we will show the primary endpoint. We will probably, as a little extra, the cherry on the whipped cream, we will share some interim results of our palmoplantar pustulosis study. So I would say in the near future, watch out and see if all this excitement around nanobodies, if this is all converting into really better outcomes for diseases that currently have a significant unmet need.
Kristian, thanks for joining us on Beyond Biotech.
Thank you so much, Dylan.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.